Good morning, my name is Julissa Rubio Soto, and today I'm excited to share with you my summer research project on diurnal and nocturnal microbial concentrations in the air. Before telling you more about my research, I'd like to ask you a couple questions. Does anyone here suffer from antagonizing allergies? And if so, do they seem to be worse during specific times throughout the day? Personally, I notice that my allergies seem to be worse during the night, so I wonder, what is in the air during the night that causes my allergies to seem worse? In order to answer my question, I dove into the field of aeromicrobiology. But what is aeromicrobiology? Aeromicrobiology is actually the study of movement, passive transport, deposition, and dispersion of bioaerosols in the air. And what I learned was that air is a major mode of biological transport, and that microbes in different environments make up the aerobiome. I also learned that some microbes can elicit or aggravate allergic responses, such as asthma, atopic dermatitis, and COPD. Given that my allergies seem worse throughout the day, I wanted to know, do microbial bioloads in the lower atmosphere vary between day and night? So I hypothesized that microbial bioloads are higher during the night due to cooler night temperatures and the absence of UV light. In contrary, I hypothesized that microbial bioloads are higher during the day due to human and animal movement. So in order to answer my question, I sampled the air. On top of the chemistry building at CU Boulder, using an air sampler that was pulling in air into a filter at a rate of 2 liters per minute for approximately 12 hours. From 7 a.m. to 7 p.m. for 8 consecutive days through mid-June. After collecting my air samples, I went back to the lab and used culture-dependent and culture-independent microbiological methods to culture, grow, and quantify microbes. In order to quantify my microbes, I took my filters, cut them in half, and washed them to release my microbes from our filters. Then I used culture-independent DNA extractions to extract my DNA from that wash and target the 16S rRNA gene to obtain our bacterial cell copies. And that's how we gather our bacterial cell copies per cubic meter of air. For my culture dependent, we actually culture our microbes in a 1:2 dilution ratio. And after we had diluted our samples, we plated them in TSA and R2A medium. So after 8 days of incubation, I went back and counted how many colonies have grown in each plate, and that's how I was able to determine my colony forming units per cubic meter of air. So, was there a difference in microbial bioloads between the day and night? What I observed from my data was the bacterial gene copies during the night were 3 times higher than my bacterial gene copies during the day, and that this difference was significant, as illustrated here by my box plot. Furthermore, my colony forming units showed that while the night samples had more variation than my day samples, these were not significantly different. Additionally, I added some environmental metadata to see if there were any trends between what was going on in the environment and my concentrations of bacteria and fungal. And what we observed was that for my figure A, I looked at temperature over a sampling period, and the background in the figure actually represents our 12-hour segments between the day and the night of each day that we sample. Unsurprisingly, we noticed that temperature decreases during the night and increases throughout the day. I also looked at humidity to see if there was a trend with this, and what we noticed was that humidity consistently increased during the night and was lower throughout the day. So ultimately, what we found was the bacteria bioloads were significantly higher during the night. That the absence of UV radiation, cooler night temperatures, and humidity likely contribute to higher bacterial bioloads. And the humidity may provide a reservoir for bacteria. And the reason why I think that that is possible is because humidity is water vapor in the air. And what I believe is happening is that these microbes are using this water vapor as a source of reservoir that allows them to survive in the atmosphere and disperse further throughout. And that is why we saw a higher abundance during the night instead of during the day. So going forward, I would actually like to be able to take this research and do it again to different seasons. I wanted to know if during your classic allergy seasons, so during the spring, if we will see the same results or if we will see a bigger difference between those results. I also would like to be able to go back to my cultures and identify where we are actually able to grow to see if there were any known allergens in our samples. I would like to thank the RACS program for this amazing opportunity, the National Science Foundations for funding this amazing opportunity, the entire Fierer lab because they were so great to me. Everybody was so helpful and encouraged me. And I would personally like to thank my mom for babysitting. Thank you.